does it do that? That's the question. Anybody else? Okay. So what directs this? I said that we have these consensus sequences that essentially define, or partially define, and I'll show you what I mean by partially later, but partially define the cleavage sites. And we also said that all you have to do is put these things together in order for that this hydrolytic attack and then the transesterification to occur. The question is, how do you put them together and what's involved in this mechanism? And the answer is that there are little molecules that are called SNRPs, SNRNPs, SNRNP. And that stands for small nuclear. They're small, they're found in the nucleus, RNPs, ribonucleoprotein particles, which essentially means that all of these little SNRPs that are indicated by these round guys there contain both an RNA molecule and a protein molecule. That's why they're called, or at least one of each. That's why they're called ribonucleoproteins, because they have both protein and RNA in them. Okay? SNRPs in the nucleus. Each of these guys contains an RNA molecule, and the RNA molecules are between 100 and 200 nucleotides. And you can see that each of these little SNRPs is called U something. The reason they are is that these RNAs here are rich in uridines. They have a lot of the nucleotide or base uridine in them. And that's how they were first discovered. So the uridine rich, they're 100 to 200 nucleotides, and they were numbered just in the sequence that they were discovered, U1, U2, U3. So the U1 SNRP has a uridine-rich RNA and a bunch of little proteins. These things are there's tons of these, right? They're involved in processing out introns, and there's a lot of introns being processed in the nucleus of the cell. So in the nucleus, of all of these, there's about 100,000 copies of each of these that are involved in cutting out introns. So there's a lot of it going on. So these guys come together, as are shown in here, and as they set up on the splice site, right? So there's the intron 1, or exon 1, exon 2, and the intron in between. As they set up here, they form a structure that's known as a spliceosome. Somes are different than organelles. They're molecular structures within a cell, but they don't have a membrane. So they're not organelles, they're somes, ribosomes, spliceosomes, replisomes. They're all just structures with multiple enzymes and proteins, but they don't have membranes. So this is called a spliceosome that's there. And the spliceosome is made up of all of these different SNRPs. Each of those SNRPs, I don't see it on here, but each of those SNRPs, also aside from this RNA, has about 7 proteins that are associated with it. And if you count, there's 5 of those guys right there, so that's 5 times 7, there's 35. It turns out there's other proteins involved, and there's about 50 proteins, including the proteins that are in the SNRPs that are required for splicing. So each splice site requires the participation of 50 different proteins. And those proteins are involved in aligning all of these loops and everything and so forth. Okay, now, the construction of the spliceosome begins much as we talked about transcription factors. These SNRPs add in a very specific way. For instance, you can see the very first thing is that the U1 SNRP recognizes the 5' end of the first exon, or actually the 5' end of the intron. So that conserved nucleotide always there is recognized by this SNRP here. It binds. Once it does, then in an ATP-dependent fashion, the second one, the U2 SNRP, binds on there. Once those two are bound there, then in an ATP-dependent fashion, you can see there's a trimer of SNRPs. The SNRP 4, 5, and 6 come in there. And as they do, they knock out U1. So U1 is the one that localizes and finds that 5' site, and eventually these 3 are loaded on the 5' site. U2, by the way, you notice where it's sitting in here, is the guy that recognizes that branch point A. So the branch point binding protein, right, finds the consensus sequence and recruits SNR U2 to the site. So there's a branch point binding protein that recognizes that branch point A, very important. And that recruits U2 to the spot. Once U1 and U2 have recognized or identified those 2 spots, then these other guys come in and bump out this guy. Immediately, as soon as that pretty much happens, you can see see that what happens is that U4, this is not quite right. Oh, no. Yeah, well, that's weird, because this arrow kind of implies that these all come off, they don't. But U4, just this one here, comes out of the complex, here's U4, right? And it leaves U5 and U6. So that the final members of the final SNRPs that are in the spliceosome are U2, U6, and U5, right? And those are the guys that are going to actually do the moving. So U4 and U1 are pretty much involved in loading all of this on there. They're kind of like loading complexes. Are you still with me? Once these are here, you can see what happens is that the one end is cut, it begins to form the lariat. The lariat then interacts on this, or this is the lariat here, whenever it's interacting there, and it's going to form, it's going to cut that out. And as these guys are ligated together in an ATP-dependent fashion, these guys are released. In an ATP-dependent fashion, the lariat is released. In an ATP-dependent fashion, these guys are reprocessed so they can be used again. ATP, 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 over and over again. Okay. So does anybody have any idea why there's all that ATP? Can you guess? Can you guess what the function of these, what are these SNRPs doing? Why are SNRPs involved here? Well, once again, we're talking about recognition, right? And specificity of recognition. We've talked about DNA recognition. We've talked about DNA, RNA, right? Complementarity. We've talked about DNA and proteins, amino acids and bases that give us specificity. So now what we have, right, is we have this intron. We have these 3 consensus sequences, and they have to be recognized by something. So what recognizes those sequences? What recognizes those sequences in the RNA? Well, there are RNA binding proteins. There are a lot of RNA binding proteins in biology and in cells. But in this case, the recognition is done with RNA. So the recognition between this SNRP, the U1, and that consensus sequence there is RNA-RNA recognition. So it's the U1 RNA that's in the SNRP that does that recognition. Right? The complementarity between that consensus sequence, remember the consensus sequence at the 5' end of that intron? The recognition between that consensus sequence and the U1 RNA is what lines that SNRP up right in the right place on that site. Right? Everybody see that? So as I'm going to show you in a minute, all of this movement here, I'm going to begin to show you. I'm not going to show you much of it, and I want you just to barely understand it a little bit. But everything that happens in this whole processing step, right, has to do with those U1 RNAs moving with each other, recognizing the consensus sequences in the intron, and recognizing each other. So each of these RNAs in those SNRPs recognize each other and the intron. And during each step here, there's kind of like a movement of the RNAs. They relocate themselves. They react with different parts of RNAs. They interact and form complementary with different RNAs. And as they're doing, what they're doing is they're whipping that precursor mRNA around. So the precursor, right, as it interacts with all these RNAs, is essentially being moved around. Why is it being moved around? Well, it's being moved around for exactly what we showed here. What it's doing is all of these SNRPs, right? So the spliceosome is forming here. And what it's doing, because it interacts with the RNA here, the RNA there, and the RNA there, is it's lining this all up. So the function of the spliceosome is to take this precursor and to sort of bend it around and whip it around so that this guy there is very close there and it can do the biochemistry. And then this guy here is very close there and it can do the biochemistry. And the way that the SNRPs do that is with RNA recognition and interactions. Does that kind of make sense? I'm going to try and show you a little bit here. It's not the easiest thing in the world to do, but I'm going to try. This one's easy. This is from your text. This shows the RNA that's from U1. 200 nucleotide RNA that's called U1. It's the U1 SNRP. Here is the, right, this is your consensus sequence. There's the branch point. That's where the G is going to bind. This is actually the intron, or this is the exon, and the gray part, or the gray and the brown, I don't know why they do all these colors, but the gray and the brown and the green here are the intron. So this is the 5' side of the intron. That's the GU that's always conserved in the intron, right at the consensus sequence that I showed you at the very beginning at the 5' end. And here, look at the sequence in this RNA. So in the U1 RNA, there's a sequence that is complementary to this guy here, and that sequence can form hydrogen bonds. So this guy in the U1 recognizes where that branch point is, and it does so by this complementarity. So this is the picture. This is the RNA of the SNRF that's bound right here. Okay, and that's how it finds that spot, is through that complementarity. That's pretty easy to see. This is a cartoon from another book that shows the same thing. Here's the U1 that recognizes that part of the exon. Here's the branch point binding protein, BBP, that recognizes the branch point that has branch point A in it. And then the BBP will bind to U2, right? So here, once the branch point binding protein binds, right, then it interacts with the U2 SNRF, and we showed that in your other cartoon where the U2 SNRF was there. And it has complementarity to the branch point, right? And so and that complementarity is what lines it up. That and the branch point binding protein helps to line that up. And it's important, you notice there's 1, 2, 3, 4, 5, 6, 7, close. Actually, an RNA duplex is stronger than an RNA DNA, is stronger than an RNA and a DNA, so it takes less than 7 nucleotides to make a fairly stable complex with RNA. But this is right there at the border. And the reason it is because this stuff all has to move. It can't be so tight that the splices, that the RNAs can't move along. Okay, so this ought to be pretty easy. Everybody ought to see this one. Here comes the confusing one. This shows you, now look at this, so it gets